Start by saying thank you to the organizers for inviting me to this beautiful country. And I'm going to give you a brief introduction to mitosis, and in particular the problems that we're trying to understand or solve in mitosis. And then I'll go into some of the ways that we're doing this using live cell imaging. So the key question, or the key problem that mitosis has to solve, is to ensure that when the cell separates, each of the two daughter cells receives an equal and identical complement of the genome. And what you can see is that this cell is waiting until the last chromosome attaches to the mitotic spindle, which will pull the two sister chromatids apart. So it's only after the last chromosome attaches that you get sister chromatid separation, and following that, cytokinesis. So this is key to genomic stability, because if cells divide too early, before all the chromosomes are attached, then you'll get chromosome loss and chromosome gain, and that leads, of course, to aneuploidy. So how does the cell solve this problem? It does it through regulating the stability of two proteins which are inhibitors of anaphase. So there's a protein that's present in the cell, and while it's there, the protease that cuts the cohesin complex that keeps sisters together cannot be activated. So the cell cannot separate its sisters. And then there's a second inhibitor of cytokinesis and mitotic exit. And so what the cell does is it regulates the stability of those two proteins. And those two proteins, the inhibitor of the protease is called securin, and the inhibitor of cytokinesis and mitotic exit is the major mitotic cyclin CDK, cyclin B1. So the cell regulates the stability of these two proteins according to the behavior of the chromosomes. The way in which those proteins are targeted for destruction is through ubiquitin-mediated proteolysis. So the proteins are recognized through what's called destruction boxes, or Ken boxes, and they are ubiquitinated by the combination of a UBC enzyme, so that's an enzyme that carries the ubiquitin, and the ubiquitin ligase, which is a large complex called the anaphase promoting complex, or cyclosome. Now, those two together are not sufficient to degrade the substrate because the APC requires a third protein, which is a coactivator called CDC20. And CDC20 is really crucial to the activity of the APC because it's needed both for substrate recognition and to catalyze the activity of the ubiquitin ligase itself. And the reason why I'm emphasizing CDC20 is because that's going to be the target of the checkpoint that prevents the degradation of securin and cyclin B. So it's only when CDC20 is able to bind to the APC that the APC becomes active and recognizes its substrates, causing polyubiquitination and degradation. So in order to understand this process, we need to be able to, especially a process as dynamic as mitosis, we need to be able to look in living cells in real time. And so for a while, we've been making use of fluorescent reporters of the activities that we want to study. In this case, in order to look at APC activity, we have knocked in a fluorescent protein into one of the alleles of cyclin B1, and you can see that here. You can see that 50% of the protein is labeled, and by labeling the protein with by fluorescence, we can then simply film the cells in time lapse And what you can see is that as the cell goes into mitosis, top left, you'll see that cyclin B1 moves into the nucleus. Next one is the bottom of the screen. You'll see it goes in, cyclin B1 goes in the nucleus, binds to the spindle, and then is degraded. And then the third cell does the same. So simply by measuring the amount of fluorescence in the cell, we can tell how stable the protein is, and we can get an indication of when it starts to be degraded and how fast it's degraded. And because it's a live cell process, we can then perturb the cell and see if that changes the proteolysis of the protein. And by doing so, over the past few years, we see activity. We also want a live cell assay to look at the checkpoint. So again, we've knocked in a fluorescent protein into one of the alleles of one of the checkpoint proteins, in this case MAD2. So again, you can see that 50% of the protein is labeled. And when we do that and look by time lapse, what you can see is that as the chromosomes attach to the microtubules, MAD2 leaves the kinetochores, and you can see that that one kinetochore just there had more difficulty than others in attaching to microtubules. And it's only when all of the MAD2 finally leaves the kinetochores that you can then get cystochromatid separation and anaphase, and mitotic exit. So we have an assay for the checkpoint, we have an assay for the APC. So now let's put them together. So here we have a double knock-in cell in which we've got cyclin B1 labeled in green, MAD2 is labeled in red, and we also have an ectopic marker for the kinetochores, and in this case the centromeres, in blue. What you'll see is that as that cell enters into mitosis, the nuclear envelope breaks down. You have lots of unattached kinetochores, so MAD2 will bind to them, and the red plus blue is magenta. And then as those kinetochores attach, you'll see the MAD2 leave, so the centromeres will turn back to blue. And so what we're looking for is the time at which cyclin B1 begins to disappear compared to when the last of the MAD2 has left the kinetochores. So I'm just going to step through the movie, and what you can see here is the envelope was broken down, so you have lots of kinetochores bound to MAD2, so you see lots of magenta kinetochores. You can see cyclin B1 on the spindle poles, and as those kinetochores bind to microtubules, you see the MAD2 beginning to leave. So those kinetochores are turning back to blue again. And very quickly, these are normal diploid cells, RPE cells, very quickly they sort themselves out. And you can see there are only a few kinetochores now that are magenta. So most of them have attached to microtubules. And then finally, we get to this point here. We just have 2 or 3 kinetochores labeling. And then at that point, basically, all of the MAD2 has left the kinetochores, so they've all attached to microtubules. And now I want you to look at the staining of cyclin B on the spindle pole, and you can judge when that begins to decrease. And basically, it decreases in the very next frame. So what we can see, or what we found, is that as fast as we can image without bleaching, which is 1 frame per minute, then within 1 minute of MAD2 leaving the last unattached kinetochore, cyclin B1 has begun to be degraded. So it means that APC and the checkpoint are very, very closely coupled in time. Furthermore, we can actually take a cell where it's begun to degrade cyclin B, and we can reimpose the checkpoint with a spindle poison. And you can see that as soon as you add the spindle poison, within a minute or so, you have stabilized cyclin B. So here we're just measuring the fluorescence. Actually, in this case, it's securin. So that means that you very quickly turn on the APC after you've turned off the checkpoint, but you can very rapidly turn the checkpoint back on and immediately turn off the APC. And that's the question that I want to try to answer in the rest of the talk. So in other words, how can this checkpoint rapidly inhibit an APC that's already degrading its substrates. And the reason why that was a difficult question to answer is because we know, or we knew, that CDC20 requires exactly the same motif, and we called it the killer motif, to bind to MAD2 as it needs to bind to the APC. So these CDC20 binding is mutually exclusive between MAD2 and the APC. So if you're going to turn the checkpoint back on again and turn off the active APC, you would have to dissociate the CDC20 and bind it back to MAD2, and that's supposed to happen at unattached kineticals. But David Morgan had shown that it takes several minutes for CDC20 to dissociate from the active APC. So those kinetics don't match. I've just shown you can turn off the APC within a minute. So how can you do it more quickly, and how can you do it without having to dissociate CDC20 from the APC? And the answer is that it turns out that this checkpoint complex, the MCC, generated by unattached kineticals, is itself an inhibitor of the active APC. And I'm showing you schematically now because it's a little confusing, because I'm going to be talking about two different CDC20s. There's the CDC20 sequestered to make the MCC, and then there's CDC20 that's bound and activating the APC. So how do we come to this conclusion? Daisuke Izawa did this by first making recombinant MCC in baculovirus, and that's the silver stain gel to show you. And then he did a ubiquitination assay with the APC, and ubiquitination assays look like this. You can see the original substrate down here. You add the APC and ubiquitin, and you get ubiquitination. That appears as a smear. And then you can add increasing quantities of the inhibitor, and you see that you reduce the smear. And what Daisuke then did was an order of addition experiment. So he took the APC and added CDC20 and the APC at the same time, or he pre-activated the APC with CDC20 and then added the MCC. And what you can see is that you get exactly the same inhibition. It doesn't matter whether you add the MCC before or after CDC20. So that means that the MCC must be able to turn off active APC. So how does it do this? And what Daisuke noticed was that if you look at BubR1, one of the components of the MCC, it has two of these motifs that are recognized by the APC. It's got a D box and a chem box. It has another chem box here, which I'll mention right at the end of the talk. So Daisuke wondered whether those are acting as pseudo-substrate inhibitor motifs. And so he mutated them, and what he found was that he could bind the MCC to a second molecule of CDC20, but it required both of these motifs, and that's quantified on the right. Furthermore, that was essential for the checkpoint, because if we do an experiment in which we take the cell, deplete it of BubR1, and then put back either wild-type BubR1 or one of these mutants, you can see when you deplete BubR1, the checkpoint is ablated. Cells can't stop in mitosis in the presence of a microtubule poison. You put back wild-type BubR1, they stop for hours. But if you mutate this this D box, you see you hardly manage to reimpose the checkpoint at all. But potentially there's a snag to this, because if the MCC only recognized CDC20 through these D and chem boxes, it would be just as good at binding free CDC20 as APC-bound CDC20. So is there a way in which you target the MCC specifically to CDC20 that's activating the APC, because that's the entity that you want to inhibit. And it turns out there is. And this came through a parallel set of experiments that Barbara Di Fiore was doing on a motif that we had identified that's conserved between the checkpoint proteins and cyclin A, one of the other major mitotic cyclins. And we called this, for reasons that I won't go into, we called it the Abba Motif. And what Barbara found, in collaboration with Norman Davey, a bioinformatician, was that there are two of these Abba motifs flanking this Ken box that Daisuke had shown was important for the checkpoint. And this cassette of an Abba motif, a Ken box, and another Abba motif is one of the most conserved features of the checkpoint family. It's found in plants, animals, and fungi. So we wondered whether the Abba motifs might be how you are targeting the MCC to CDC20 bound to the APC, because Abba motifs bind to CDC20. And sure enough, when Barbara did this experiment of depleting wild-type BABA1 and then putting back a BABA1 in which we'd mutated either of these two Abba motifs, you see that you don't restore the checkpoint with this mutant, and you hardly restore the checkpoint with that mutant. So what are they doing? And it turns out that they are indeed essential for the MCC to bind to the APC, because here we have wild-type BABA1, and here we have this first Abba mutant, and you can see that BABA1 binds to make the MCC, MAD2, BAB3, and CDC20, but it can't bind to the APC at all. And if we do this with the second Abba motif, you can see that we also have a dramatic effect on APC binding, but it's not quite as strong as that first Abba motif. We also noticed that the spacing between the Abba motif, the Ken box, and the second Abba motif is conserved. So we wondered whether that was crucial. And sure enough, if you make the spacing too short between the Abba motif and the Ken box, you see that you ablate the checkpoint completely. But there isn't actually any particular sequence in there that's important, because you can just put back a linker of any sequence and you restore the checkpoint. So we have an Abba, Ken, Abba cassette, which has to be particular distances from each other. They're CDC20 binding motifs, and they are crucial for how the MCC binds the APC. So we postulated that they must bind the two molecules of CDC20 onto the APC, and then sure enough, in parallel with us, David Barford, whose structure this is, solved the structure of the MCC bound to the APC. This is CDC20 binding and activating the APC. This is CDC20 that's in the MCC, and this is BABA1. And this is the first Abba motif, this is the Ken box between it, and this is the second Abba motif. So it stretches around those two CDC20 molecules in a lariat formation to stabilize the MCC on the APC. So I'll leave you with our conclusions. Unattached kinetochores catalyze the production of the MCC, and that is BABA1, MAD2, sequestering CDC20, but that complex itself can then bind to a second molecule of CDC20 and stabilize it in what's called a lariat formation. So I'll finish with the people who did the work. So Daisuke Izawa on the right, Barbara Di Fiore in the middle, and they were helped by Oksana Nash-Chekina. And I'm funded in particular by Cancer Research UK, and thank you for your attention.